name is Vaishevich Albert, and I want to tell you about one interesting mechanism occurring in cells of living organisms, more specifically in mitochondria. Each of us, like this hamster, feel warm. But not every one of us has asked oneself why we are warm. Where does heat come from?
Mitochondria take part in the process of cell respiration.
Part of the energy from food eaten by hamster goes to produce heat. In usual mitochondria, it happens like this.
After the preparatory stages of sugar metabolism, namely glycolysis and Krebs cycle, the reduced molecules NADH, NADPH, FADH give the electron to the ATC, electron transport chain, and the realized energy is used to move protons to the permitochondrial phase where those creating a transmembrane gradient of protons concentration, nu H+. This gradient is further used for ATP synthesis and production of heat.
However, I want to tell you about another unusual type of mitochondria. There, a very unusual process takes place. It is called uncoupling of electronic transport with phosphorylation. In usual mitochondria, these processes are coupled with each other. The phenomenon of uncoupling of electronic transport with phosphorylation was shown when studying a compound which later was called uncouplers. The uncouplers do membrane permeable for protons H+ or other anions, so they reduce the mu H+ gradient or completely liquidate it.
Couplers make holes in the internal membrane of the mitochondria, so in the presence of uncouplers the ATC works in vain. In such conditions of the ATPase stops synthesizing ATP and electronic transport goes with the highest speed.
In this case, electronic transport is uncoupled with ATP synthesis and is called uncoupled. Uncoupling of different types presents an extremely convenient tool for research of energetics of membrane. Usually, the function of uncoupling is carried out by small hydrophobic molecules, easily soluble in the lipidic bilayer.
Some of them, for example antibiotics, gramitidine or nonactin, build in the membrane and form channels that protons can pass through. In other cases, uncouplers form a mobile complex with an anion. The bi-negative or positive charge is removed from the membrane. Protonophores, being weak hydrophobic acids, cross the membrane in the protonated form and remove the pH gradient.
Uncoupling of electronic transport with phosphorylation plays an important role in the metabolism of plants and animals. The point is that, as I already told, besides the energy storage in the form of nu H+ of ATP, one important function of cell respiration is dissipation of energy in the form of heat. With high probability, I can assume that everything said before is not completely clear for people who are not well familiar with biology or functioning of mitochondria in particular. Let's imagine there are some students and some teachers in the class. Students are the reduced molecules. Having prepared for the exam, students have information that is a pool of protons. Teachers are ATA synthesizers. Teachers watch the students without giving them a chance to cheat. But they force the students to think, giving them the right directions. Teachers promote to synthesize ATP, that is a knowledge. The process happens in the usual mitochondria. When there are more students in the class and the same number of teachers, each student will not watch. There are students who have an answer K, a grip, that is an uncoupler. The grip does not allow the pupil to think themselves. The students will choose the easier and faster way of copying the answers from the answer K, thereby generating heat that is receiving a good mark for the exam. But they do not synthesize ATP, that is a knowledge, since there are not enough ATPases for everyone and the way of copying seems more attractive. The same process occurs in the mitochondria of the brown fatty tissue, which is present at the animals which fall into hibernation. The energy of nu H+ is used not for ATP synthesis, but is dispatched in the form of heat warming the body of animals or some bodies of plants responds to cold. A good mark increases the average score of the class.
dispersion results from increase of proton conductivity of a membrane along with the general increase of fatty acids. Circulating through a membrane, they are capable of having the uncoupling effect. In the premenstrual phase, the anion of a fatty acid connects the proton and moves it into the mitochondrial matrix, where reverse dissociation occurs. But to make the system work, it is necessary to return the anion of the fatty acid. For this purpose, there is a carrier.
It can be projected onto our improvised experiment. Having cheated, the pupil pays an uncoupler to another pupil. Meanwhile, his hands are a carrier delivering the uncoupler to new protons, the information of other pupils. As plant cannot increase the temperature of all their body, they need a mechanism of uncoupling for other purpose, namely regulation of electronic transport. Against the background of excess of ATP, plants need to oxidize NADH quickly without ATP synthesizing. In our class, there are persons who are filled with knowledge, ATP, to such an extent that they simply need to do the test the oxidation of NADH quickly and protest to their own affairs. And as we are just learned, the fast way of the solution of this problem is cheating, uncoupling. At home, they prepared some answer case. At the test, they quickly swap their sheet with their answer case. The second function of uncoupling in plant organism is protecting against an oxidative stress. An electron, having paid the ATC, moves to the oxygen molecule. In the cells, molecules of superoxide radical start accumulating. It is quickly neutralized by superoxide scavenger, forming a hydrogen peroxide. In this condition, it is possible to reduce a formation of a superoxide radical if the transport is increased and the chain is reloaded through restoration. These molecules constitute a danger to the cell, since they are strong oxidizers. At high nu H+, breaking an electronic transport promotes restoration of a redox enter in a chain. What dangerous oxidazes can appear in our class? In our class, there are so-called D students who are at the bottom of their class. Before the test, they managed to get a couple of bad marks, molecule of superoxide radical and hydrogen peroxide, which fall between for the grade points average of the class, and D students in particular. To avoid the decrease of the grade point, the disruption of membranes and other bad processes, pupils resort to the good old method of cheating, uncoupling, receiving a good mark for the test. Those many processes in cells seem extremely difficult, but after the detailed consideration they stop being so frightening and becoming so fascinating that you could sort them out every evening instead of viewing tiresome telecasts. Today, I have tried to explain you the process of uncoupling of electronic transport with phosphorylation, which plays an important part in both plant and animal mitochondria, including ants, in our infancy period, and I hope that I managed to do it.